I'm going to talk about chitosans, which are derived from chitin, as you know, and they are a class of polymers which have fascinated us now for more than 25 years, and we've been trying to unravel some of the secrets of structure-function relationships of these interesting polysaccharides. As you know, chitosans are derived from chitin, which is very abundant in the biosphere. And chitosans are probably among the most versatile and most promising functional biopolymers that we have. They can be used in very many different areas for different applications. And for us as biologists, of course, life science applications are most interesting. And as plant biologists, we're mostly concentrating on agricultural applications, where, for instance, it is known that when you treat plants, as you see here with groundnuts, when you treat plants with chitosan, then you may have a better yield and also good protection from disease. But as I said, you may have. This is the problem. Well, it used to be the problem with many applications of chitosans, that sometimes they work and sometimes they did not work. And of course, based on such an unreliable activity, you cannot really develop any products. So we started early on to say we need to understand structure-function relationships. We need to know which chitosan is responsible for which bioactivity if we want to come up with reliable applications, for instance, in agriculture, but of course also, for instance, in biomedicine, where you can use chitosans for wound treating, and under such chitosan dressings, even deeper burn wounds can heal without the formation of any scars. So if it works, it is really miraculous, but the problem is it doesn't always work. And so that's why we are focusing on structure-function relationships. So very briefly to remind you, of course, in this audience, I do not have to introduce chitin and chitosan. I just want to remind you that chitin is, due to the acetyl group, is a rather hydrophobic polysaccharide, crystalline and insoluble in water. So this is difficult to work with in biotechnology, but when we deacetylate chitin, if we completely deacetylate it, we generate polyglucosamine, and then we have the free amino functions, which at slightly acidic pH values will be protonated, and so we give positive charge to the molecule. And this polycationic molecule is then also hydrophilic and soluble in water. Of course, the most interesting chitosans for us are the partially acetylated chitosans. So those which contain or which still contain some acetylated hydrophobic residues, but also some deacetylated cationic residues. So you end up with a polymer that is partly hydrophobic and partly polycationic. And this, of course, matches well the most other macromolecules and polymers in biological systems, which are also partly hydrophobic, but then partly polyanionic. So the distribution of hydrophobic patches and polycationic sites will determine the interaction of chitosans with a lot of other molecules. And this is what we are trying to understand. So first, initially, we decided to concentrate on two structural parameters, which are the degree of polymerization, so the number of units within the polymer, and the fraction of acetylation, so the number of residues which still carry an acetyl group. So together with chemists, we teamed up with chemists who would then generate for us chitosans with different degrees of polymerization and different fractions of acetylation. And we came up with this, what we call the chitosan matrix, in which we have on the x-axis the fraction of acetylation. So we have glucosamine on the left side and acetylglucosamine on the right side. And on the y-axis, we have the degree of polymerization. So we go from monomer to oligomers, to large oligomers, small polymers, and finally large polymers. And of course, in the upper right-hand corner, we have the insolubility range. This is chitin, hydrophobic, crystalline, not soluble in water. But all the remainder of this matrix is accessible to us. There we can have soluble chitosans, and the bioactivity of these and the solution properties of these can be analyzed. So together with the chemists, we generated a series of chitosans which are derived from a single chitosan, for instance, from this one, which is completely deacetylated and has a degree of polymerization of around 100. And then this chitosan is chemically N-acetylated to yield a series of chitosans which differ only in the fraction of acetylation. Or we take this chitosan, which is a high molecular weight chitosan with a high fraction of acetylation of around 40%, And this is partially depolymerized to yield a series of chitosans which differ only in the degree of polymerization, but have constant fraction of acetylation. Now, using such chitosans, we can perform bioactivity assays. And as I said, we are plant biologists and mostly interested, therefore, in plant strengthening activity of chitosans, but also in direct antimicrobial activities, which of course are also relevant if we want to develop chitosans for plant protection. To this end, we first had to develop quantitative assays. So we have a quantitative assay for the antimicrobial activity in which we mix a certain amount of this, a fixed amount of bacteria or fungal spores with different concentrations of chitosan. And then we simply measure the optical density over time. And then we can see that with increasing chitosan concentration, the growth of the microbes is reduced. And this then gives us a quantitative measure of the antimicrobial activity of different chitosans. On the other side, we have one example of many in which we measure bioactivities in plants. These are wheat leaves, and we have injected, using this small device, we have injected chitosan solutions into the intercellular space of these leaves. And then if after 2 days we decolor the leaves, we can see that there is a reaction has occurred. And here we have injected a series of chitosans with increasing fraction of acetylation. And we can clearly see that a completely deacetylated chitosan is completely inactive. You have no reaction, just like after the injection of water. But when we increase the fraction of acetylation, the reaction becomes stronger. But then if we increase even further, the reaction goes away. So if we come close to chitin with a fraction of acetylation of 0.7, then again, the bioactivity is completely gone. This was absolutely important result for us, and I like this picture because it makes very clear, it makes very visible that it matters a lot which chitosan you take. If you take the right chitosan, you can get a strong reaction. If you take the wrong one, you get no reaction. So of course it matters which chitosan you take if you want to have a specific reaction. So doing such kind of assays, many different assays on many different plants with many different chitosans, we finally came up with this, with the bioactivity matrix for chitosans, in which we can see that there's an area of rather small polymers with very low fraction of acetylation. This is a high charge density. They are best for antimicrobial activities, whereas the chitosans which have strongest plant strengthening activities have a higher molecular weight and also a higher fraction of acetylation. And based on these understanding of structure-function relationships, we were then, of course, able to come up, for instance, with this product, which contains just chitosans, a mixture of chitosans which have these activities. And this is now something which is reliably working year after year to really protect plants from disease, not completely, but to an extent that the farmer can be sure to have a good harvest, even in the presence of a pathogen. And if there's no pathogen pressure, then the treatment of the plants with this chitosan leads to growth increase. So the yield is then definitely better than the non-treated plants. This is an important aspect because it makes farmers spend money on the product, even if they don't know yet whether in this season a pathogen is going to occur or not. So based on this work, we were quite content with what we achieved for plants, but, and this is actually now true for many other areas of research and applications as well. Of course, we were not the only ones to find that if we can control the degree of polymerization and the fraction of acetylation, then we can come up with chitosans which have reliable structure-function relationships, reliable bioactivities, and then they can be used for the development of products. And there are now a number of industrial producers of chitosans which have good quality management, which can produce such second-generation chitosans. And this is now leading to a renaissance of chitosan-based products. And for the first time in the history of chitosan, the demand for chitosans, for high-quality chitosans, is higher than the production capacities in the world. But of course, as scientists, we're never content. We're never at an end of our questions. So the next question we asked was, after working a little bit on nanoformulation, which I'm not going to talk about, we were interested in the question of the pattern of acetylation. Does the pattern of acetylation influence the biological activities? So the distribution of the acetyl groups along the polymer chain. Why do we think this should be important? Here we have 3 different chitosan polymers, just in theory, which have the same degree of polymerization and the same fraction of acetylation, but the acetylated units are differently distributed inside the polymers. We have a random distribution here, we have a blockwise distribution in the middle, and on the right-hand side, we have a regular alternating distribution. Now, if you imagine that in a tissue to which you apply this chitosan, you would have a sequence-specific chitosan hydrolase. Like this one, this would be lysozyme. In our body, we have lysozyme, which has chitinase activity. It can cleave chitin and chitosans, but it requires 3 acetylated units next to each other for a single cut. Now, if you imagine that this blockwise chitosan would be degraded, then the acetylated blocks would be degraded to small polymers, where the deacetylated blocks would remain intact. If you would have this one, which has a regular alternating distribution, you would never have 3 acetylated units next to each other. This would not at all be degraded. Whereas the typical chitosan, which you can buy today, which has a random distribution of the acetyl groups, would be degraded to a rather broad mixture of small and large chitosan oligomers with different random patterns of acetylation. So if a target tissue contains a sequence-specific chitosan hydrolase, and we now know that almost every organism produces chitinases or chitosanases, which are sequence-specific, then we should expect that the pattern of acetylation has a strong influence on the biological activity of the chitosan. Now, the problem is that today's chitosans are typically they are produced using chemical ways, and then they have a random distribution of the acetyl residues. So we would have now a 3-dimensional chitosan matrix in which we add the pattern of acetylation as a 3rd dimension. And we can determine the pattern of acetylation, for instance, using 13C NMR, where we can also calculate a number, the PA number, which we can indicate here. PA1 would indicate a random or possibly random distribution. And if the PA value is below 1, it tends to be a blockwise distribution, and above 1, it tends to be a more regular distribution. But as I said, when we generate chitosans using chemistry, by partial chemical D-N-acetylation of chitin, or by partial acidic depolymerization, or even by partial acetic N-acetylation, chemical N-acetylation, we would always remain in this 2-dimensional matrix. We would always have random pattern of acetylation. This is not bad, because we know what we have. We know we have random patterns of acetylation. But if we want to find out whether the pattern of acetylation really has an influence on bioactivities, then we need to control the pattern of acetylation. And our hypothesis was that using enzymes, it should be possible to move out of this 2-dimensional matrix and add different non-random patterns of acetylation. So for a long time, we focused on identifying such enzymes, and we used different knowledge base or also unbiased discovery programs to look for such novel chitin and chitosan modifying enzymes. We developed techniques to identify them, to characterize them, and all of this with the purpose to finally use them to produce chitosans with non-random patterns of acetylation, which we could then use in different applications to find out whether the pattern of acetylation really has an influence on bioactivities. So we start with chitosan hydrolases. Here we have a chitosan polymer with a fraction of acetylation of 0.35, so 35, 1/3 of the residues are still acetylated, and as this is produced chemically, it has a random pattern of acetylation. Now, this is degraded by 2 different recombinant enzymes, bacterial chitosanase, which requires 2 deacetylated units at the -1 and -2 position. And then if such an enzyme cleaves this polymer, you are going to end up with a mixture of oligomers, but they are partly defined now in terms of their pattern. They are end defined. The reducing end is defined. It will always have 2 deacetylated units. On the other side, we use, again, a bacillus chitinase. This one requires an acetylated unit at the -1 subsite. So that we will get a different mixture of products, and they will all have an acetylated unit at their reducing end. So we now have mixtures of partially acetylated chitosan oligosaccharides, which are partially defined in their pattern of acetylation. And now we can use these mixtures to determine their bioactivities. And Y assay, which we use, is a simple elicitor activity in which we treat suspension cultured cells or also intact plants with a potential elicitor active fraction. And then we measure the oxidative burst, so the rapid and transient production of hydrogen peroxide, which is induced within a few minutes, and after about half an hour, it's over again. So this is a very simple assay for elicitor activity of compounds such as chitosan. Now, when we use a chitosan mixture produced from this chitosan polymer with fraction of acetylation 0.35%, which is produced by acid hydrolysis, and therefore the oligomers have random patterns of acetylation, then we can see that we get a very clear increase of the bioactivity of the elicitor activity with the degree of polymerization. And we see that the smallest elicitor active oligomer would be the nonamer, DP9. Now, this is what we see for random pattern of acetylation, mixtures of chitosan oligomers with random patterns of acetylation. Now, if we look at our oligomers, which we produced using chitosanase or chitinase, we can see that the chitosanase products, which have the two deacetylated units at their reducing end, they are strongly elicitor active, whereas the chitinase products, which have the acetylated unit at the reducing end, they are inactive as elicitors. So very clearly, the pattern of acetylation at the reducing end is important for the biological activity, for the elicitor activity of these chitosan oligomers. Now, the second assay we were interested in was an assay for priming activity. A priming agent would be something that is recognized by the plant, which is triggering the plant to increase its capacity for signal transduction. So when in the next step an elicitor is seen by the plant, then there's more signal transduction, to put it simple, and there's a stronger resistance response. So priming is like vaccination. You treat the cell, it doesn't yet perform a resistance response, but it is prepared to perform the resistance response. And when the elicitor comes, the response is even stronger. So again, we can do this in our assay. In this case, we pre-treat our cells with the priming agent, and then depending on the system we have, 2 or 3 hours later, we treat it with the low concentration of elicitor, which by itself induces almost no oxidative burst. But if we have a pre-treated cell which has seen the priming agent before, we see a very strong increase in the burst. So this is our priming activity. We use a low concentration of elicitor, which by itself is almost inactive in a naive cell, but in a pre-treated cell, we get a strong response. Now, again, we can do this with our enzymatically produced chitosan oligomers. And again, we see the chitosanase products are priming active. The chitinase products are not priming active. So we know the chitosanase products were elicitor active. Now they're also priming active. Next, we tried to find out, we looked for the dependence of this bioactivity, priming activity on the degree of polymerization. Again, we see that with increasing degree of polymerization, the priming activity increases, only for the chitosanase products, not for the chitinase products. But interestingly, we also see that the DP4, the tetramer, has a clear priming activity. Not a very strong one, but there clearly is a priming activity, which is clearly stronger than for the larger oligomers. It's as strong as for the DP8, for the octamer. Now, when we analyze this in more detail, then we can see that at low concentrations, the DP4, the small oligomer, is priming active. The DP4 is priming active, but it is not elicitor active. The DP12, the larger oligomers, they are both priming active and elicitor active, but the small one, the DP4, is priming active without being elicitor active. This is very important for us because this would be really a vaccination of the plant. No, it would not trigger the resistance reaction in itself. It would only prepare the plant to trigger the resistance reaction once an elicitor is seen because a pathogen is there. So we were analyzing the chitinase and chitosanase tetramer in more detail. Here you can see that we separated it by chromatography. So in the chitinase product, we see a D2A2, so a tetramer which is twice deacetylated, and a tetramer that has 3 deacetylations, only 1 acetylated unit left, Whereas in the chitosanase product, the one that is priming active, we only see a monoacetylated tetramer. Now, of course, using mass spectrometry, we can analyze this further, and we see that the D2A2, the double acetylated chitosan or double deacetylated tetramer in the chitinase product, is a mixture of these two. And of course, we see that as expected, they always carry the acetylated unit at the reducing end. Then we analyzed the monoacetylated tetramers, and as expected in the chitinase product, the single acetylated unit is at the reducing end, whereas in the chitosanase products, the two units at the reducing end, as expected, are deacetylated, but we have a mixture of the two tetramers which carry the acetyl residue at the non-reducing end or next to the non-reducing end. So now we have analyzed the priming activity of these tetramer fractions, and then again we can see the chitosanase tetramer, which is the mixture of these two, has priming activity, whereas the chitinase product with just a single acetylated residue at the reducing end is inactive as priming agent. Now still we have a mixture of two, and we wanted to know which one of these two really carries the priming activity. And now we use the second set of enzymes, which are chitin deacetulases. In this experiment, we use two different bacterial chitin deacetulases, not B from Rhizobium and VC-CDA from Vibrio. And they are specific in, they are regioselective in that they deacetylate specific units. Not B only deacetylates the unit at the non-reducing end, whereas the Vibrio enzyme deacetylates only the unit next to the non-reducing end. Now, when we use these two enzymes, we can knock down one of these tetramers. For instance, using the Vibrio enzyme, we can convert this one, the second one, which carries the acetylated unit next to the non-reducing end. We can completely deacetylate this one, but leaving the other one intact. Not B does the other way around. It leaves only this one intact, but it deacetylates this one. Now, what we see is that in both cases, the activity decreases, but both of these mixtures are still active. So it looks like both of these oligomers carry priming activity, whether the acetylated unit is at the non-reducing end or next to the non-reducing end. But of course, we still have mixtures here because we also have the fully deacetylated tetramer, even though we know this one by itself is inactive. Of course, it could be that there is some interaction between these two. So we wanted to have fully purified, fully defined oligomers, and this can be achieved using these regioselective chitin deacetulases because we do not only have these two. One thing is we can combine them, so not B can act on the product of VC, CDA, and the other way around. So we can generate oligomers which are deacetylated at both of these residues at the non-reducing end, or we can use fungal enzymes which have a different mode of action. They can typically deacetylate all units within an oligomer except for certain units they cannot attack. For instance, the one from this wheat rust fungus cannot attack the two units at the non-reducing end, or this one from the fungal endophyte Pestalozziopsis can also not deacetylate the reducing end. Now again, we can combine these fungal enzymes with the bacterial enzymes and create even more different oligomers. So with a little more than a handful of well-defined regioselective chitin deacetulases, we can now generate all 14 tetrameric Bacchus. We can also prepare all pentamers and quite many of the different hexamers. Of course, we can only produce very small amounts of these for the time being, but even small amounts are good enough for, say, bioactivities. And I'm just showing you now the activity of the priming activity of the monoacetylated tetramers. That's what we were interested in. And here you can now see that if we use these fully defined monoacetylated tetramers, we can very clearly see that the priming activity completely depends on the position of this acetylated unit. The closer it is to the non-reducing end, the higher is the priming activity. So what we can very clearly say is, yes, patterns matter. Different patterns of acetylation will have different bioactivities. So this was our key question at the beginning. Does the pattern of acetylation determine bioactivity? Now that we can control the pattern, we can say, yes, it does. The idea behind this now is, this is what we are trying to understand now, for instance, using RNA-seq. approaches that a fully acetylated chitin oligomer that is recognized by the plant as an elicitor. This tells the plant there's a pathogen, so it has to stop all activities, it shuts down photosynthesis, and it starts defending itself. On the other hand, a priming agent is only telling the plant that a pathogen may be there. So the plant keeps going. It even boosts photosynthesis, but is prepared for defense. This explains why a plant treated with the right chitosan will have even better yield if there's no pathogen pressure, because photosynthesis is increased in preparation for defense. If there's no defense, then the extra energy provided by photosynthesis can be used for growth. So clearly we are able to control the pattern of acetylation in chitosan oligomers. But can we do the same for chitosan polymers? This of course is a lot more difficult, but we have started to work on this also. We used our chitin deacetulases on polymers. We quickly found, like others had also found, still finding that chitin deacetulases are almost inactive on crystalline fully acetylated chitin. So we need to do a first deacetylation step using chemistry. Then we end up with a highly acetylated chitosan which has a random pattern of acetylation. And then this one can be used as a substrate for different types of chitin deacetulases. And typically these enzymes will not fully deacetylate the polymer, but they will stop at a certain lower fraction of acetylation. Here we have stopped the enzymes when the products reached a fraction of acetylation of 0.3. And then we can compare these chitosans with the chitosan produced using chemistry, where we know that the pattern of acetylation is random. We can do this using enzymatic mass spectrometric fingerprinting and PCA analysis. And then we can very clearly see that the chemically produced chitosans, which have a random pattern of acetylation, are clearly different from the enzymatically produced ones, which apparently have a different pattern of acetylation. And they also differ between the three enzymes. So depending on which enzyme we use, we get different patterns of acetylation. And we now know that the PES CDA, for instance, gives a more random pattern. The Protospora CDA gives a rather even distribution of the acetyl groups, whereas the Rust enzyme gives a more blockwise distribution of acetyl groups. When I say here random, it doesn't really mean it's random, because we can clearly see here that even for PES CDA, in PC1, there's a difference between the chemically produced ones which have random PA and this one. So this is closer to random than the other ones, but it's still not random. So using these enzymes, we can now for the first time control to some extent the pattern of acetylation of chitosans, and we can generate a PA series of defined chitosans. So these chitosans all have more or less the same degree of polymerization, a little bit more than 1000, the same fraction of acetylation of around 30%, but they differ in their pattern of acetylation. Of course, what you can see here is that of the potential space of different patterns of acetylation, our products only cover about half of it. So we cannot access, using this strategy, we cannot access the full range of the pattern of acetylation. And that's also not surprising. We cannot really reach blockwise distribution because our substrate with the fraction of acetylation of 0.6 already has a random distribution. So there are no large acetylated blocks in such a chitosan. So we have continued working, and we've now come up with a different way of producing chitosans, again using our enzymes, but in a different way. And we can generate chitosans even with a very strongly blockwise distribution. So here we can extend now this series of chitosans towards the blockwise pattern of acetylation. And we can do the same, of course, also with the chitosan with a lower fraction of acetylation. So this one is a chitosan with a fraction of acetylation around 0.1. So only about 10% of the units carry an acetyl residue, but these few acetyl residues are distributed in a blockwise fashion. And I'm going to show this in one of my last slides, which gives more biomedical application of this chitosan. So we have used this chitosan for transfection of human cells. And we can see here, so we have used the chitosan to create nanocapsules into which we have added DNA for the green fluorescent protein. And then we can see that in a negative control with no chitosan, there is no transfection. If we use a positive control with lipofectamine to help the DNA get in, the mRNA to get into the cells, we can clearly see transfection. And we can see that this chitosan, which has a low fraction of acetylation, but a blockwise distribution of the acetyl groups, has an even stronger or equally strong transfection activity as lipofectamine, which you can also see here. This chitosan, low fraction of acetylation, around 0.1 only, but the blockwise distribution of acetyl groups is a very good transfection agent. To show the difference between these chitosans, we have digested them and using our fingerprinting technique, using different enzymes where we know the sequence specificity. So we have either used a chitosanase or different chitinase or lysozyme. And the interesting thing you see is that the blockwise, this chitosan with the blockwise distribution of the acetyl residues, even though it has a very low fraction of acetylation, is a substrate even for lysozyme. So lysozyme, which requires 3 acetylated units next to each other, is able to degrade a chitosan produced in this way, even though it has a fraction of acetylation of 0.1. If only 10% of the groups are acetylated, then in a random pattern, you would very rarely have 3 acetylated units next to each other. So with a random pattern of acetylation, you see no products. But in our case, with the block distribution, we can clearly see products. And when you look at this in more detail, then what you can see is that we even have an A4, a fully acetylated tetramer. And if you think about the way this enzyme cleaves, then to generate this tetramer, you need 2 additional acetylated units at the non-reducing end and 1 additional one at the reducing end. So this is a heptamer. You need 7 acetylated units next to each other to produce the A4, the fully acetylated chitin tetramer. With a fraction of acetylation of 0.1, 7 acetylated units next to each other is almost impossible. But because we have this blockwise distribution, this chitosan can be degraded and can yield the A4. Why is this important? The problem with using such an application for chitosan, for instance, if we would want to use chitosan for the application of an mRNA-based vaccine, then the problem would be that this mRNA would be taken up by the cells, but it would not be degraded inside the cells because the human enzymes like chitotriosidase or acidic mammalian chitinase or lysozyme are not able to digest such a chitosan, but they are able to digest a low, this block PA chitosan, which has good transfection efficiency. So this, I think it's a breakthrough result that we can use chitosan internally. Even if they are taken up by human cells and they are taken up easily, they can transfect human cells easily, they can deliver drugs easily, but they are degraded in the human system, and so this removes a hurdle for their pharmacological uses. So in the end, we can again say patterns matter, and I think that with this control of the fraction of the pattern of acetylation, we can move from second generation chitosans towards third generation chitosans, which would be even more defined, would have non-random defined patterns of acetylation. We should know not only their structure function relationships, but also their mode of action. And I'm convinced that such chitosans, which of course now are only available on lab scale, will create new opportunities for the development of chitosan-based applications in the future. With this, I'm ending my talk, and I thank all of you for your interest and for listening. But of course, most of all, I'd like to thank my group, because all the work that I've talked about, all the results that I have presented, have not been generated by myself, but by my excellent students. With this, thanks.
Thank you so much, Bruno, for a really, really nice talk. I'm really happy to see that the field is moving towards this third generation of chitosans. It's really a huge progress for this polymer. So now the floor is open for questions. Well, if no one has, I have a couple. Where you saw the transfection efficiency, was this into live cells, and then you think that the reason for the block where you have low FA and block PA, that there are either a chitinase or lysozyme that cleaves the chitosan and then releases, or is it the chitosan itself that sort of is better in transfecting?
I think it's both is the case. If you look at here, we in gray, we have the chemically produced ones. So even with the chemically produced chitosans, a fraction of acetylation of 0.3 is less effective than 0.1. So the low fraction of acetylation, the more positive charges are better, most likely because they are better taken up by endocytosis into the cell. But those which have the block PA, they are even better. So they may be taken up even better, this we don't know yet. We know that both of them are taken up well. We think that probably the release from the endosome is better because these chitosans are digested inside the endosomes by chitotriosidase, and then this contributes to the release of the cargo.
So do you have an explanation for, let me see, do you have an explanation for the differences you observe on pattern of acetylation for the priming effect? So what is the reason for why one is more effective than the other? Do you have any ideas on that?
I think the most likely explanation is that there should be a specific receptor which is responsible for this bioactivity. If you have a tetramer in which the bioactivity is so clearly dependent on the distribution, on the pattern of acetylation, I think a receptor is the most likely explanation. We are hunting this receptor, but we don't have it yet.
Okay. Now there's a question from Peter. You can unmute yourself, and Peter West, and you can unmute yourself and ask your question.
Yeah, hi Bruno, thanks for a very nice talk. I was wondering, do you do anything along the lines of enzyme engineering to modify or optimize the specificity of these enzymes that change the acetylation, and then hence perhaps facilitate large-scale production of these molecules?
Absolutely, of course we are doing this. Huh? We are engineering our enzymes to change the regioselectivity, or to make it even more strict, so that we have that we get the kind of products that we want to have. But of course, another goal would also be to have enzymes which are better produced. These enzymes actually they're not easy to produce, but once if we have the recombinant enzyme, they're very stable. But of course, we are interested in scaling up the production from milligram amounts to gram amounts, which that's ongoing. I guess that we can probably also scale up to perhaps to a kg amount. But I would guess that for agriculture, for instance, for agriculture purposes, this would not be the way to go. This would be too expensive. But for biomedical applications, I could imagine that production of such chitosans with defined patterns of acetylation is possible in an industrially relevant scale.
Thank you.
There's a question from Olivier Tranquit. Thank you for your talk. What are the plant receptors able to discriminate priming tetramer from elicitor one?
That's the question which of course, that's a highly important question. We know that for instance, this tetramer with monoacetylated tetramer is not active, is not recognized by the typical chitin receptor. So in plants, chitin receptors are known, but we know that they require a rather high fraction of acetylation to bind the chitosan oligosaccharides and also to be triggered by them. So the receptor which we postulate to be there for this priming activity is not yet known, but it must be a different one from the known chitin receptor.
Excellent, good question. Any other one who has a question? Pedro has a question.
One for Pedro, yeah.
Yeah. Do you want to ask it yourself, Pedro, or should I do this?
Hi, can you hear me?
Yes.
Yes, we can.
Some GH7 enzymes have chitosan degrading abilities. And I just wonder if you had studied any one of them in this context, or and if you have any ideas on their application.
It's definitely an interesting question. It's known in literature that chitosans can be degraded by enzymes that are not typical chitosanases, for instance, cellulases, but also some lipases and proteases. And we have so far never used them. We have not tried this. It would be very, I'm a bit skeptical sometimes, a lot of older publications at least, which used enzymes purified from some microbial source, they may not have been completely free of chitinase activity. So some of the reports which say that lipase is active as a chitosanase may actually be residual chitinase or chitosanase activity contaminating the enzyme preparation. But when using recombinant enzymes which are well characterized, it is still possible apparently to degrade chitosans with such enzymes that are probably not meant for degrading chitosan. We have not used them, so we don't know whether they would also show sequence specificity and would thus generate specific chitosan oligosaccharides, which would be interesting. It's a very interesting question, but we have so many chitinases and chitosanases to work on that we have not yet ventured into other enzymes.
Okay, thank you.